Okay, thank you. What I'm going to present today is very much the viewpoint of developing phages to get them out to as many patients as possible. So, what do we have? We have stages of development. We have a regulatory path. We have ways to actually get them into the clinic, and we have the way to take them forward and make them usable by the largest possible number of patients. The stages of development will be very familiar to some of you, less so to others, so I will run through them. You have preclinical. This is a huge body of work, everything from molecular target selection through to advanced in vivo animal models. And this is really the body of work which you put together to take to the regulators to say, we think this can go into people, we think there is evidence that this will be safe, we think there are indications that it will be effective. And this does need to be assembled in a way which the regulators can then cope with. That is followed by a phase 1 safety trial in the classical pathway. A phase 1 trial is a small group safety trial focused on metabolism, focused on the way the drug is actually processed, excreted, used by the patient. That is followed by phase 2 trials, where you go into another small group, but this is a small group with your target indication. These patients give you preliminary data, again, on safety, but also on whether your candidate therapeutic is doing what you think it should be doing. Following that are what are called pivot trials, phase 3 trials. These give you large group safety and efficacy data. Normally, you will need 2 of these run independently. However, for some orphan drug indications or where there are reasons to believe you simply can't access the number of patients, you can go with 1 of these. A phase 3 trial can be anything from a few tens of patients, if you've got a really difficult orphan indication, through a few hundred, up to tens of thousands of patients. Now, the very largest phase 3 trials, unsurprisingly, cost hundreds of millions of dollars or euros. But there is something of a lack of understanding over what the costs of the early stage trials are. These can be done for numbers in the low millions of dollars, or sometimes even lower. It is possible to get through this program without the kind of telephone numbers that people like to suggest is necessary to make this progress. We know because we've done it. But underlying all of this is CMC, 3 very important letters. Chemistry, or in our case biology, manufacturing and control, which includes all quality control, quality management, making sure that the product you start out with will be able to go all the way through the process and come out the other end and be used. If you decide not to bother with this, you'll get a certain way through the process, then you'll have to stop and start again. And that isn't a good idea. So you need to be aware of the requirements all the way through for this. And of course, even after you're on the market, you can do additional trials for other indications or for issues the regulators still want resolved. But all the way from phase 1 to phase 4, you need to be aware of the quality of the product. CMC is actually a major cause of delayed and denied market applications. People can get all the way through to putting something together, wanting to sell it, having their trial data, and then discover they can't. You have to progressively build the understanding of your product, and any major changes can result in you being told it's a brand new product, start again. You don't want that to happen. And of course, there are some unique development challenges with phage. First, we know that the phage host range depends on the host bacteria. You have to work with paired phages and hosts. The phage can, of course, co-evolve with a host bacteria, both in vivo and in vitro. You have to control your quality through your production process. The phage genome is mutable. The shared virus may not have an absolutely identical sequence to the product. How do you control for that? What limits do you set? You have to define those with the regulators. And we already know that when we find a new phage, most of the genes will be completely unidentified. We won't know what they do. Some, yes, but most, no. You have to be aware of that. And of course, some phages can be unstable. So that restricts your shelf life if you don't select those out early on. And of course, all of the above are part of what has to be considered for the CMC process. Now, the regulatory path. Individual countries have their own regulators, and the levels of regulation are extremely variable. There are still countries in the world where there is very little regulation, but those tend to be countries where there isn't a particularly large market for the product, although there may be a need for it. This is just a list of examples. In Australia, we have the Therapeutic Goods Administration. In Brazil, the Sanitary Surveillance Agency. China has got their own FDA, who have a very different policy towards rooting out issues within the FDA, which on occasion involve shooting the person who heads it. That hasn't yet been followed in the US. In Europe, we have the European Medicines Agency, but that is an overarching organisation above local national agencies, such as the MHRA in the UK. And you can still go through those national agencies, what's called the decentralised process, as against going through the core EMA process, which is required with certain types of medicine. So it all gets very complicated. Then in India, in Switzerland, and of course, coming up last, the big one, the FDA in the US. Now, existing regulations, those that are already out there for other kinds of product, provide guidelines to a development pathway. Appropriate interpretation and effective navigation of those existing regulations is necessary. The regulators want to be certain things are safe. They don't change things because they like us. They can't. Early and open discussion with the regulatory authorities is essential for success. We have done that from the get-go, and we have found them to be extremely helpful. Companies developing phage therapeutics will have to comply with existing regulations. Those regulations put safety first. Efficacy is important, but it comes later. Uncontrolled or anecdotal studies are not relevant to the US and EU regulators. So what have we been doing all this time? Nearly 100 years now. Well, since 1919, it's been clear that there is therapeutic potential for phage. Commercial phage products were made and sold in Europe from the 20s onwards, some even after World War II. But they were very dubious quality. If anybody knows where to find the report that Durrell tested 20 preparations and found not one phage in any of them, I'd like to see it. I think it's an urban myth, but it's a compelling one. Certainly we know that the quality control in those products was just not up to the job. And then phages were often used where there was no bacterial disease. And even where there was, people thought there was the bacteriophage, one bacteriophage that ate all bacteria, which of course is completely wrong. We know they're highly specific, but just to give you an example. This is an advert from the 1920s, medico-biological laboratories in the United Kingdom. You will see that bacteriophage therapy, it doesn't say what type of bacteriophage, is indicated for eczema, which is an allergic condition, herpes, which is caused by a virus, urticaria, another allergic condition, and my personal favourite, gallstones. Now, if anybody here can suggest to me how a phage might have an effect on gallstones, I would be fascinated. Okay?
Some gallstones are from bacterial origin.
But once they formed?
What?
Once they formed?
Once
Some are caused...
Once they formed, diffusion is done passive inside. But you cure the cause.
The cause, yes. But once they form, once they're symptomatic, that's not going to happen. So, as you can see, you clearly had a lack of understanding of what bacteriophages were and how they could do it. Yes, urticaria can have a bacterial origin, actually. There can be bacterial involvement in the early stages. So, yes, but once it's symptomatic, no. So, phages, we have a number of things we have to do. And yes, there are virus products on the market, but many of them are grandfathered. a rather peculiar term, which just means they were approved many years ago. And there are certain levels of tolerance given to grandfathers which aren't given to the rest of us, often involving unpleasant noises at the dinner table. But we do not have the ability to say we want that level of tolerance. These things are out there, they've been out for a long time, and certain things are tolerated. The classic one is aspirin, which would fail any clinical trial it went into today. Current regulations for new drugs are a lot more stringent, and the regulators are hyper-vigilant about safety. It has to be proven, not assumed. The label claim, what you're actually saying your product will do, the label is critical. It's just the term referring to that. Has to work around current antibiotic standard of care. You can't say, I'm going to take this patient off antibiotics, they're not really working and I'd like to try our product instead. You have to work with the existing standard of care. In some areas like burns or leg ulcers, that makes it very complicated because you can't take those patients off existing therapy. There are some areas where antibiotics have just ceased to work, where you can, but you have to bear that in mind. And the regulators want quality by design. This means that when you design your drug, you work to build in quality from the start. The problem is we don't go and design our drug, we go and find it in various environmental sources, which I won't go into. But you can't build in quality by design when you're picking up something which has existed for 1 billion years. So you have to have some way to think about that, to answer the question, where is your quality by design? The approaches to clinical use. Millions of patients are being treated, many of them in the bacteriophage inquiry in British India. But that data is not transferable to the US and EU regulated environments. To date, fewer than 100 subjects have been treated in the handful of regulated clinical trials which have been published. Clearly, that is not enough for approval anywhere. In order to realise the promise of phage therapy, we must conduct well-controlled clinical studies conforming to current regulatory standards. Now, there are approaches to this. You can go with predefined phage mixtures. Empirical therapy, the patient has the infection, give them the mixture. And that's based on a mixture shown to be effective against a high percentage of the target bacteria. Or you can go personalised medicine, pret-à-porter or couture, to quote, where you match the phage, the bacteria from the individual infection. Now that requires companion diagnostics, which are complex and expensive. If you are looking at companion diagnostics, you start out with the patient. They have an illness, you think it's bacterial, so you take a specimen, you send it off for microbiological examination. That gives you a bacterial isolate. That then has to go to a specialised phage lab, which has the capability to test susceptibility. You then match the phage to the isolate from your bank of approvable, usable phage. GMP, good manufacturing practice. Oops. You provide your cocktail and it's used to treat the patient. Now, it's a fairly complex flow, but it is key that at every stage what is sent is what arrives. It is documented that what is sent is what arrived. And if asked several years later, you can verify that what was sent is what arrived. There are a lot of acronyms here. GCP, GLP, Part 11 compliance, CLIA, HIP, AA. These are all various sets of regulations that apply to make sure that is the case, and they all have to be done. And there are unique clinical challenges. The standard of care I've already mentioned. That means that your initial studies may be limited to situations where a small number of patients, antibiotics are just not useful. That's what we did in our phase 2 work with late-stage ear patients where antibiotics didn't work, so they were treated with ear hygiene. That meant we could just go in with cleaning of the ear without the conflict of concurrent antibiotic use. And that does limit the number of patients. There are a lot of patients out there with burns or ear infections or leg ulcers, but how many of those can you actually get into the trial? That was mentioned in the last presentation. The final product presentation has to be a factor in ease of use of both patient and clinician. How does the patient expect to be treated? It's been an issue, for example, with the use of maggots in medical use, although they are now in use. And what does the clinician expect to use? How does that work? Personalised medicine and mixture use may require sensitivity testing. You may need companion diagnostics, because one thing phages are, we now know, is highly specific. There is no point giving them if the target isn't there. You can't just say, you've got a bug, have a phage, unfortunately. And of course, you have to be sure that what you're giving actually gets to where it's needed. If you're treating ear infections or topical infections, that's not a problem. The phage goes onto the wound, the phage goes onto the burn, the ear. But if you're delivering it to the lung, or even more, through the blood, that's an issue. It's got to get there, but it's got to still be active when it does. So, what do we do from here? We need to determine acceptable global regulatory strategies. If you just want to go for prescribing in one small geographical area, that may not apply, but that's not what we're doing. We need to do something which will be used everywhere. You need to manufacture the product under appropriate good manufacturing practice. Now, what does that mean? You have to agree that with the regulators and manufacture your product accordingly. This is part of the CMC process. You have to establish what your product actually is. You have to conduct well-controlled, adequately powered clinical studies to unequivocally confirm safety and efficacy of the phage in the defined target indication. You have to determine the appropriateness of the study design and what you're actually measuring with the regulators and clinicians. There are protocols out there which are used for existing medicines. At present, we have no choice but to use those protocols.